Hi everyone, just great pleasure to be here. Amazing city, amazing food, amazing city. Well, I'm talking about the conference, of course. Okay, so thank you very much. We love to study superpowers in nature, and one of the superpowers is organisms that are resistant to cancer. And what we try to do is try to understand how we can use these species that are resistant to cancer to maybe better understand cancer and apply it to human. Okay, so cancer is all over. 1 in 2 men will get cancer. 1 in 3 women will get cancer. But it's not only us, of course. If we look at Nature, recent paper published in Nature show that cancer is actually all over metazoan. But it's not only metazoan. Now, if you look at plants, okay, so what you see here is a tumor that happens spontaneously in plants, and here is some tumors that happen as a result of some pathogens. And this is something really cool. Look, here's a corn, and you see what's happened to one of the seeds when it's infected with a fungi. Then it's become from this beautiful yellow thing to this disgusting gray thing filled with amylase. So this fungus that moved to Europe from the Americas with the corns are actually people here in Europe try to eradicate the fungus. They barely succeed. Do you know what people doing with this fungus in the America, or what Mexicans are doing? Do you know what they're doing with it? So they eat it. That's the way to treat your tumor. Okay? So anyway, it seems that cancer is unavoidable part of nature. And why is it like that? Why it's so hard to fight cancer? Because of course it's a disease of the genome. We know that the tumor evolve and adapt based on Darwinian evolution. So when we give drug and kill most of the cells, there's many that survive. And of course, because of the mutation, because of the diversity, it's actually a group of diseases. So it seems that cancer is avoidable, but this is not true. Cancer is an avoidable part of nature, and actually, cancer is already solved in nature. It's not solved once, but it's solved multiple times, and nature has the solution. And genomic, we are here in genomic and evolution. Genomic data is the key. Now, let me show you some of the species. Of course, many of you know them that are resistant to cancer. We spoke before about whales. So whales are huge. They have 1,000 times more cells than human, million times more cells than mice. They live up to 200 years. So the chance of whale cells to become tumorigenesis is 1 compared to mice is million times lower. And if you are talking about elephant, then they have 10 times more cells than human, but only 5% cases of cancer. Again, their cells are much more resistant to cancer. And this ugly fellow here, which is the naked morat, one of the ugliest mammals that exist. It's basically almost totally resist to cancer. It's very, very hard to induce cancer in the naked morat. Okay, so there are multiple studies that try to understand how these species are doing that. You know, for example, in the naked morat, they show that there is a dense extracellular matrix. They compare it to other rodents. In elephant, there are multiple copies of p53. By the way, most of them have pseudogenes. There's leaf pseudogenes again. There's whales, we're talking about better DNA repair, low metabolic rate. And in Spalax, this is how do I call it? It's the Jewish friend of the naked morat. It actually lives in Israel. Anyway, so it has improved P16. But what is the problem? So this is, of course, a fantastic idea to try to compare all these species and try to find what make them resistance to cancer. But when you compare one species to the other, then what you show that there is the solutions are very hard to understand. So we need a lot of work. They're very hard to study. Of course, elephants are not the ideal lab animals. So it's very, very hard to put animals in the lab. And they're, of course, very hard to apply to human. Okay, so that's one problem. The second thing is when you compare one species and find one solution that exists in the species, you don't really sure that that's actually related to cancer. It might be related to the fact that elephants have trunk, or that this organism, as we said before, is really, really ugly. So that's another thing. And another very important thing, that even if you find something that's important to the naked mole rat to be resistant to cancer, it doesn't really mean that it's relevant to human. Okay, it might be really toxic to human if you do the same solution, or not affect at all. So the questions that we had is there any universal anti-cancer mechanism, and can we find it, and can we apply to human, and how it should look like? So what should we search for? Okay, so we recently published a paper that showed that if you look across all genes, across all the different species that are resistant to cancer, you see that many of the protein correlately change or evolve more in organism resistance to cancer. But we looked for something different. So we want to identify, or what we try to look for is the smallest and most specific changes. So we want to compare the prone species to the resistant species and try to see if there's even a single amino acid that might be different. Single amino acid in single protein that might make this protein better in resistance to cancer or in the species. And why is it? Why does it really even make sense to look at such small changes? And I think we had 3 different guidelines. The first is simple. Think about the situation. You have a mutation that happened in a single species of entire, out of all the other species, out of all the elephants, one of them get this mutation. The chance that it will happen is depend on how complex is this mutation. So point mutation is much likely to happen. But if you think about taking P53, duplicated, change many things, then the chance is, of course, lower. And remember, it needs to happen multiple times. Now, the second thing is that, of course, after it's happened, then it has to be with it need to improve the fitness, because without that, it will not distribute along the population. And the third thing is global. So we should see the improvement of fitness potentially across all these different species. Okay, so simple, high fitness, and global function. If we find things like that, then it has some other advantage, but I will talk about them a little bit later. So here is an example. Okay, here a specific protein. We look at all these rodents that are prone to cancer, and then we see that here's a specific amino acid that change. Arginine, it changed to arginine. This is the naked mole rat from before. Then we look at other rodents, also resistant to cancer. Also, it has arginine instead of histidine, and so on. So in all the rodents, all these rodents that are resistant to cancer had arginine. Now, we did the same thing and look for whales. And again, here, all the whales have arginine instead of histidine, and elephants as well. Now, the question here in this specific, for this specific protein is, what about humans? Are we resistant to cancer or not? So the answer is that it depends. So we are much better than, of course, to mice and other species, but we are not perfect as well. So in human, what we see that most of human in this case has the cancer-resistant variant. Now, most of humans, but what about the other humans, the less lucky one that have the prone allele? So what we found is that if you look at the prone allele, the people that have the prone allele, have 10 times more chance to be among the cancer patient, and also in this mutation in this specific region appear in this specific cancer. Okay, this is an anecdotal example, but I think it's very important. So what we did is we developed an algorithm. We screen all these different species, divide them to the resistance one, to the prone one, and look for a location where all the resistant species have a specific amino acid compared to the prone species. You know, we did it, we score each one of these positions and so on. I'm not getting into the details. In the end, we found close to 200 positions like that along the genome. Here are some examples. You see here's 100% the same amino acid, here's a little bit less. So just to give you some idea of what's happening. So what's happened in the end is that we have a map of all this region where we have this anti-cancer positions. So what we want to do now is, of course, to understand what the chance to get this number randomly. And of course, we did a permutation. We found that the signals is 8 times more than what you expected by very random. Another very interesting thing is we look at all this cancer resistance variant across many other species that we didn't, including the one that we didn't test it. So what you see here, their size, the weight, and their lifespan. Of course, if you are big and live longer, then it means that you are resistant to cancer. You have to, because if not, you will die young. Okay, and what you see here is that all the one with the brown, with the black dot contain most of the variants. All the species that are short-lived and small, or most of them, have almost none of these variants. Here you see the naked morad. You see bats that are small but live much longer. You see that are much more black compared to what you expected. Another thing that we did is we screened the TCGA and try to see what's happened in all the cases when we have SNPs. So some of the human have the cancer resistance variant and other have the cancer prone. Of course, it's not happened a lot, but in the cases that we found, we see that in what you see here is that, and we divided to different population, is that in many cases, the resistance allele appear much more if you compare between healthy and patient. Of course, it's very noisy. We need to do additional thing, but in general, this is the trend. Here is another example, and another example, and so on. The final thing that we did, we tried to validate it. So we tried to see if we can take human cells and a specific gene, in that case, LIG3, which related to DNA repair. It's one of the genes that work with BRCA1, the breast cancer cell, and try to see what will happen if we will take human cells and use CRISPR and make these cells, only one change will be changing the amino acids from the prone to the resistance. Again, we are a bioinformatics conference, so I will not get deep into the analysis. What we found is, although it was a change in a single amino acid, we find a lot of genes that changes their expression level. We show that the cancer resistance were actually less, the cancer resistance cells, the cells with the cancer resistant variant were less malignant. They had reduced motility, reduced transformation. They respond less good to DNA damage, and as a result of that, following DNA damage, they just die faster compared to the cells without the cancer resistance. Okay, so finally, so we generate this database and we find global, so the same amino acids that appear again and again in all the cancer-prone species. It's very important because it suggests that it will be effective. The chance that it will be effective, since it's the same solution, since it's convergent evolution in all these different cancer-resistant species, So the chance it's effective, it's much higher. Of course, as a result that it's not toxic, the same solution in all these species, it will not be toxic in human. That's what we predicted. And of course, it's easier to find. Look how easy is it. And of course, when it's simple, then it's much easier to understand what's happening here. Does it affect the protein structure or doing something else? So it's easy to understand, easy to recapitulate as we did. And as a result of that, it might be more relevant to human. Okay, so by that, let me skip that. Okay, so I want to take my amazing group and Lamis, who did most of the work, Dana and so on. And by that, I would be happy to take questions. I don't know. Thank you.
A couple of questions.
Thank you, it's fascinating. So it is in humans, we consider cancer as the old age disease, right? So what about these species? The age of onset of cancer, is it also like late? Because if it is the case, how come it's under selection? Because it would be after the age of reproduction.
Well, so what we think that in general, we have a lot of mechanisms that protect us against cancer. So I think that it's always a balance. So it's late onset because of this mechanism. Okay, so and there's so we have all these mechanisms that protect us, and as a result of that, you know, that it's considered to be an old age, so the evolution doesn't work with it. But it's not really true. That's why we develop all these mechanisms. I think that I confused myself. Anyway, I hope it was clear.
So basically, in the other species, then the cancer incidence is occurring at earlier ages across these other species that
the one that are prone.
Yeah.
So the one that are prone, most of, for example, mice, you don't find mice that die in nature from cancer, only in our labs. So usually they die from cold, from cats, from poisons, for things like that. Okay, so that's why we don't have selective pressure that to generate these anti-cancer mechanisms.
Hi, thank you for very great talk. I just wondered how old are these cancer-resistant variants? You do kind of phylogenetic analysis of this. If they are old variants which were lost in some species, or the basic variant was not resistant to cancer, and this resistance was something that it was later.
Okay, so I didn't have time to get into it, but I think in cases like that, okay, then probably it was convergent evolution that happened later. And what you see in some cases, it's really depend on per case. In some cases, you see that the cancer resisted, there was a positive selection. So you see that all the variants, there is a noise, or in the cancer-prone species, they have multiple different amino acids, so the location itself is not really important. But in the cancer-resistance species, you have a single amino acid. In other cases, it looks like that. So you can predict that it was T in the ancestor, and then things change over time.
Okay.